There are two words we want you to remember today. Eat methane. Okay, calm down. We're not actually asking you to eat methane. But these two words happen to be our team's primary objective, to engineer microbes to eat methane. But why? The problem. Methane is a ridiculously potent greenhouse gas, nearly 86 times more effective at trapping heat than carbon dioxide. And while CO2 is a big problem, methane is also a primary contributor to greenhouse gases, responsible for around 16% of emissions. Methane also contributes to ground-level ozone, which is attributed to approximately 102,000 deaths due to respiratory diseases per year. Back to methane. Where does it come from? It comes from all sorts of places. Natural vents in the earth, various animals and humans passing gas, cows. No, not from there. From here, cow burps. And also fossil fuel extraction. But we're focusing on one of the biggest contributors to methane, an area that is responsible for 16% of emissions. Landfills.
Let's take a moment to review how landfills work. A giant hole is dug into the earth. It is lined with clay, plastic, and geotextile fabric. Garbage is then compacted into the landfill and is layered up with dirt. This is where most people will forget about it. However, the garbage isn't actually forgotten, coats the organic matter, leading to the production of methane. The government requires landfills to control their methane emissions. In many active landfills, this is done with the process known as flaring.
We visited a landfill just, I think 15, 20 minutes away from here, and we found pretty much what we had initially been described in papers, but up close in real life. There is actually a huge machine dedicated to burning methane. You can't actually see the flame, but you can see heat sort of radiating off the top of it.
I think the vast majority of the landfills in the world are really just unmanaged dump sites. But generally speaking, methane handling is not, in many places, is not a priority, and so nothing gets done.
When landfills reach max capacity, they are covered with a plastic liner and vegetation, and they become a closed landfill. Because they have been covered for years, closed landfills emit less methane than active landfills. They also have more systems in place for controlling methane emissions. According to the National Resource Defense Council, 61% of landfill gas, which is primarily methane, is generated at landfills with no collection system, and at least 20% of this gas at landfills with no collection system simply escapes. But we think that the secret is actually in the soil.
Project goals. Our goal is to engineer Bacillus subtilis to eat methane and mix it into the soil that covers landfills, creating a biofilter. Methodology overview.
We chose Bacillus subtilis because it's one of the most common soil bacteria. On top of that, Bacillus subtilis is hardier and sturdier than normal methanotrophs, so we're more able to easily transfer it and use it in landfills instead of methanotrophs that are entirely reliant on methane.
Now, how do we go about editing Bacillus subtilis? Well, we're taking advantage of an already existing cycle called the ribulose monophosphate cycle. So this process ends up being converting methane to methanol via an enzyme called methane monoxygenase, and then methanol into formaldehyde via an enzyme called methanol dehydrogenase. We focus on the first step of this conversion during this iGEM project due to time constraints.
New composite part.
We chose to focus on using the mini SMO developed by a team in the University of Korea. It's a recent paper published in May of 2024. And the advantage of this is it's a smaller version of SMO. So it has fewer subunits, only 2, which means less things for us to insert. And it doesn't require any protein chaperones. The cherry on top is it was tested in E. coli to have shown a higher efficiency rate than normal SMO. Using this enzyme, we now need to consider how are we going to insert the DNA. Bacillus subtilis has a very convenient feature, and that's what makes it so popular for synthetic biology. It's naturally competent. So here's our full construct. We've got our Amy back and Amy front. Those are our identical little pieces that are going to match the Bacillus genome. We have spec R, which is a gene that grants antibiotic resistance to spectinomycin. We have PSPANC, which is a promoter region. we have the SMO mini alpha and beta subunits. That's the enzyme that we're trying to insert. We have a terminator region, which is a relatively standard terminator for Bacillus. And we have LAC-I, which is a repressor that inhibits gene expression. And we deactivate this repressor using IPTG. In the end, what this ends up meaning is that our gene is IPTG activated, which is very useful. So, for our genetic sequence, we need to figure out how to split up so we could conveniently order it. To this, we ended up splitting up into 4 parts. 2 of them, luckily, were actually copies of this plasmid called PDR110 that we were able to get from the University of Wisconsin. And the other 2 were sequences from the University of Korea, and those were synthetic sequences that we ended up ordering from IDT. SMO alpha failed IDT's QC, and so it ended up needing to be split into 2 more fragments, complicating things. But initially, we started out with this.
This is our DVTL cycle, that is design, build, test, and learn. In our design bit, we designed our DNA construct so that it could actually transform into B. subtilis. We had the help of our mentor, Leah McKinney from the University of Wisconsin-Madison to help revise our DNA construct.
From there, we headed to lab, trying to combine all the fragments that we got from IDT, as well as the 2 pieces that we got from PDR110. And then we attempted to transform our combined fragments that we'd combined via Gibson assembly. And guess what? We had no transformants. To troubleshoot, we tried to PCR our Gibson assembly. And then we realized a problem. Our Gibson assembly was 3,000 base pairs shorter than expected. So this meant that in our PCR product, we saw something that was entirely not our gene.
So in the second cycle of design, build, test, and learn, we started to rethink our lab protocol. We instead decided that we should start with PCR purified DNA to do our DNA assembly. Otherwise, we might have some contaminants that might disrupt our DNA assembly. So we decided to not PCR Gibson assembly to avoid amplifying the portion of PDR110 that we didn't want to amplify. We also decided to use a competent cell prep that was simpler and more time efficient.
Results.
We ran a Gibson assembly on all of those PCRs that we did, and using the purified PCR reactions. And this time, we actually did have transformants on our plates, so that was great. The bad thing was, after a few days, we actually had transformants on our negative control plates as well. We ran a starch test. We have some mixed results. Some of them clearly the starch has been being processed, but in some of them there's actually a fair amount of iodine that remains purple. And so these are all different colonies, and some of them look like they might have been successful, some of them aren't really, or are clearly not successful.
Human practices. To address this global problem, we began to look locally, reaching out to experts across multiple disciplines. We visited local landfills with our idea. We talked to the environmental manager of a newbie landfill. He showed us the flares and pipework used at landfills and their inefficiency. He recommended that we consider placing our bacteria into the biofilter, the layer of soil above a landfill. His recommendations have been instrumental in how we're building our project. We spoke with a representative from the EPA who requested to remain anonymous about government regulations of landfill sites. They noted that while larger landfills are required to capture and flare methane, smaller landfills face challenges in implementing these technologies due to limited resources. Based on this meeting, we hypothesized that our project could be advantageous for these smaller landfills. Our solution may offer a more cost-effective mechanism for methane capture that can be easily integrated into biofilters. After discussing the major issue of releasing engineered microbes into the environment, our team began developing containment strategies. We also talked to other companies working on similar projects. We talked to Ben Tarbell, CEO of UpCarbon, who confirmed the potency of methane and was excited about our approach. He introduced us to Olya Irzak, CEO of Frost Methane, who recommended making the bacteria dependent on methane, making it impossible to survive outside of the landfill's methane-rich environment, addressing microbe containment. She also was interested in future applications of our project.
I know that you originally set out to engineer this with landfills, and I'm absolutely with you that it's very promising for landfills. But there's other methane sources. So once the characteristics are there, active coal mines and manure ponds and these Arctic permafrost methane sources, like I have, a hands-on experience with those. Worth talking to like actual practitioners in the space, because then they'll be like, oh, I've had this problem. I need something with these characteristics to solve it.
When we began our work on the iGEM project, Lab School did not have the necessary lab equipment and safety gear for our work. As a result, we decided to create 3D printed and more affordable versions of mini centrifuges and thermocyclers. However, our lab facilities were still inadequate for planned experiments. Enter Counterculture Labs, which calls itself a community-supported microbiology makerspace, where science is for everyone. We met Anthony Tan, a synthetic biology expert from the University of California, Berkeley, and STEM education advocate. He helped to train our team on experimental protocols and assisted us in setting up our experiments at Counterculture Labs.
We interviewed Dr. Gould, former chair of Physicians for Social Responsibility. He mentioned how flaring methane, a popular solution, led to the production of nitrous oxide. Furthermore, the National Resource Defense Council finds that this combustion also produces dioxins, a group of perpetual toxins, and using landfill gas for energy often leads to increased pollution. Dr. Gould was optimistic about our solution as a part of a larger strategy to reduce the impact of methane emissions. Safety considerations.
What we realized was that silicate-based systems could be really effective because sand is already often applied on top of biofilters or crushed glass. Using some sodium silicate, which is slightly soluble, could be used in place of that sand. There are multiple inducible promoters, thankfully, for silicate, and we found one that allows for 11-fold increased expression of a gene when silicates are present. What this means is that the bacillus could have a toxin-antitoxin system that would only allow the bacillus to survive near the glass in the landfill and nowhere else.
Education.
The importance of education in science. Well, science is all about understanding the universe and pushing the frontiers of our understanding of the universe. But I personally think that most of us learn best from other people, learn it potentially even from our peers. And when you do peer-to-peer learning, it's not just a very scalable way of getting very personalized attention. When you explain the concepts, you tend to understand it deeper. Someone who can't explain a concept in very simple terms, I believe, fundamentally doesn't really understand that concept.
To gain an even deeper understanding of our subject matter, we decided to put Sal Khan's philosophy into practice by developing a 10-week synthetic biology course for interested middle school students.
A lot of people interested in joining the team hadn't taken any previous biology courses and didn't know many of the lab techniques that we were planning on using in our iGEM project. To correct this, we decided to teach a course at our school that would cover this basic synthetic biology knowledge. On Schnick and UV prepped a 10-week-long curriculum to cover the basics of molecular biology, as well as more advanced techniques such as PCR, gel electrophoresis, and DNA sequencing. The class consisted of 1 lecture a week, as well as 1 lab. We based the curriculum for these sessions on the community college classes we took, a collaboration with Rice University, and feedback from guest speakers. These guest speakers included Dr. Hernandez at UCSF, Dr. Vidwans at Garden Health, Dr. Cothery at Stanford. Out of the 13 students who attended this course, a majority joined our current iGEM team, showing the impact of this class. However, we didn't want to aim this class at only confirmed iGEM members. This was actually a great chance to educate the greater community about biology and introduce a lot of people who weren't sure if they wanted to join iGEM to biology as a subject. We provided all of the material for the class in our wiki for future iGEM teams and other aspiring scientists to use.
Additionally, we gave a presentation about our project in synthetic biology to Lab School 5th and 6th graders. We led them through a petri dish experiment.
I'm glad that we got to engage more with younger kids. Hopefully in the future, some of them will actually join iGEM and be able to contribute to our team in the field of synthetic biology.
There was also a community meeting at our school in which the team informed other peers about synthetic biology. 80 students attended and participated in an ethics discussion about genetically modified organisms. Overall, we approached education by making synthetic biology more accessible to students of all ages. Our courses influenced many to join our current team, in addition to educating others on uses of synthetic biology.